Uh, Speak here of Physics Club of ISRI Mohali, and will be your host today. With great delight, we would like to welcome Dr. Steffen Rulands from Max Planck Institute for the Physics of Complex Systems, Germany, to be here with us for a talk. A brief introduction about him. He is a theoretical physicist working at the Division of Biological Physics at the Max Planck Institute. His work is right at the nexus of biology and physics, where he studies the statistical physics of living systems using non-equilibrium statistical physics, data science, and artificial intelligence tools, collaborating with the experiment groups. And just before we start, a little housekeeping. Please type your questions or any queries you encounter during the talk in the comments. I'll try to take them up after the presentation ends. So yeah, thank you so much, and over to you, sir.
Yes, thank you. Thank you very much, Akash. Thanks for the invitation and the opportunity to speak. So I gather you have a very nice events coming up with a lot of interesting stuff happening this week. And I'm very honored to be giving a talk on the very first day. So let me just share the screen. Now let me know if you can't see that. The slides should be coming up. Can you quickly confirm that you see the slides? Akash?
Yes, sir, we can see the slides.
Okay, great, very nice. So in my talk, I would, as Akash said, so in my group, we're working at the interface between theoretical physics, statistical physics, and biology. And in my talk, I would like to give you an impression on how methods from theoretical physics can give insights into the principles that underlie biological systems. And at the end of this talk, I would also give you a glimpse of how biological systems, the structure of biological systems, gives rise to new problems at the frontier of non-equilibrium physics. So to start, biological systems, they consume energy from the environment. And by consuming energy, they're not in terminal equilibrium. And because they're not in terminal equilibrium, entropy in these systems doesn't necessarily increase over time. The second law of thermodynamics, if you specified just an isolated biological system, does not necessarily hold locally. And that means that biological systems are able to construct highly complex structures in space and time. And a striking example, and my personal favorite example, is the development and the self-organization of cells in the embryo. So these cells follow very complicated patterns, very complicated behaviors that interact with each other to give rise to complex organs like the brain or the heart. And in biology, there was a revolution, a technological revolution going on in the last 5 years or so, where we're now able to study such processes like embryonic development with unprecedented molecular detail. So this is an example of such an experiment. Now, this is from single-cell genomics, and what this means is that in this plot you can see on the x-axis there is the position on the DNA. So in these terms, this plot chromosome 1, roughly 200 million base pairs. And for each of these 200 million base pairs, we can measure whether this base pair, this molecule on the DNA, has a modification or not. On the y-axis, you see the index of different cells of the embryo. So for all of these roughly 600 different cells in the embryo, we can measure with utmost detail the molecular states in these cells with a resolution of single molecules. And this is an example of epigenetics, very important processes during development. And as a physicist, you can ask, it's not something we typically do. We don't measure every single spin in a magnet. We can't do that. And we also wouldn't have the idea to come up with such a measurement. It wouldn't come to our mind. Yeah. What does it actually mean that we're able to probe the detailed microscopic states of biological systems? The first, when these methods came up, actually already before that in other systems, like in social systems, with the advent of big data. Yeah, so the experiment that I just showed you has the size of the data, if you download them, has a size of roughly 1 terabyte. Yeah. With the advent of these big data experiments in biology, people said, so if we just measure everything, yeah, we know everything. Yeah, we don't need any theory. We don't need to understand things. Yeah, we just measure them. Yeah, and there was articles. There was an article like this one I'm showing here that proclaimed like the end of theory. We don't need any more theory. But is that actually true? So is it true that if we measure everything on the microscopic scales, we know already everything about a system that we're looking at? Yeah. If you look at another example, yeah, this would be the same experiment done by a car. So if you list all of the parts a car is made of, yeah, if I give you this list or I give you this photo, you would have a hard time understanding how a car works or how an engine works. Yeah, and the reason for this is called symmetry breaking. Yeah, so that means that the rules that govern on the microscopic scales don't necessarily also apply to larger scales when you put things together. Yeah, how a motor works, you can't just learn from listing all the screws the motor is made of. Yeah. Translated to physics, that means that in physics, we have already, for a long time, a theory of everything. Now, that's just the Schrodinger equation or quantum mechanics. Now, quantum mechanics is perfectly fine if you don't come close to black holes or the particle accelerators. Yeah, for normal everyday stuff, quantum mechanics is perfectly fine. Yeah, but quantum mechanics is not very, although we can solve the Schrodinger equation for small systems like an atom or small molecules, yeah, it is not very useful for understanding how a gas works, why this table here that holds my laptop is solid, and how a cell works. Yeah, although it is the correct microscopic representation of these things. And the reason for this is that the rules that govern on the quantum level, for example, in this case for water, yeah, so if you see here, I don't know if you can see the pointer. Yeah, so you have a water molecule, and this water molecule you can turn around, you can translate, you can move it around, and the wave function or the Schrodinger equation that governs this water molecule is always the same. It's invariant under these symmetries. But if you put many water molecules next to each other and you cool them down, yeah, then they suddenly build crystals, ice crystals. Yeah, and if you zoom even more out, then you get these snowflakes, these very complicated patterns. And none of this is directly in the Schrodinger equation. So we say that these are emergent properties, that the rules that govern on this quantum level, on this microscopic, the smallest level, these symmetries are broken when you zoom out and you go to the macroscopic level. Yeah, and that means that on all of these different levels here, the microscopic level and the macroscopic levels, there are different laws that emerge on these scales. And we can't just understand an ice crystal by looking at the Schrodinger equation of a single water molecule. Yeah, this is how it works in water. And of course, there are many other examples in science where this principle of emergence or symmetry breaking appears. For example, swarms, every single fish it alters its direction in these swarms here according to its neighbor. And what then arises are these complex dynamic toruses that they use to distract prey or predators. And another example here, traffic jams. Yeah, so you can have the emergence of very complex patterns in traffic, like traffic jams, just by individual driving mistakes of individual drivers. And another example is, of course, my favorite example, that's embryonic development, where every cell has identical genetic information, and this genetic information, then the symmetry between cells is broken by interactions between these cells. And then these cells collectively, through perform very complex processes, like in this video here, the onset of a very important phase in gastrulation, which is called in development, which is called gastrulation. Now, all of these examples show that behavior on the macroscopic level, if you put many atoms or cells together, cannot be straightforwardly inferred from what's governing on the level of a single cell, on a single car, a single fish, or a single water molecule. And we need tools, additional tools, that allow us to infer these collective behavior from these microscopic rules. And this is what the field of statistical physics deals with. So in statistical physics, we have developed methods that allow us to infer collective properties of systems that are composited of many individual particles. And this is also what we do in the group. In the group, we use methods from statistical physics, and in particular, non-equilibrium statistical physics, like field theory, renormalization group theory, and so on, to understand collective processes in biological systems. Some part of the group is working on how cells self-organize into tissues. We're working on how molecules collectively decide what cells should do. And we're working on machine learning, and we're also interested in how biological systems that are organized on different hierarchical scales, how they control how information flows between cells to perform a specific function. So in my talk, I will give you two examples. Namely, I will start by looking at a simple insect society to see how these biological systems send around information between different scales of organization, between the microscopic and the macroscopic scale. And then I'll go on and I'll ask, what can we actually learn from measurements on the macroscopic scale, in this case on the tissue level, about what individual cells are doing. To begin, and because this is a two-parts talk, that's very convenient for you guys, because if you don't understand something in the first part of the talk, you can always then concentrate on the second part and still get into the talk again. Okay. So in biological systems, we are usually, or biophysics, what we usually are interested in, how biological systems build complex structures. Think about the brain, the eye, and so on. So that's what we're typically interested in. But in recent years, it has also become clear that these biological systems have the capacity to break up and rebuild these structures. For example, cells perform on a very long time scales, on very long times, very specialized functions. For example, on the right-hand side here, you see the tissue of the lung. There you have different cells, and all of these cells have different jobs. For example, the blue cells here, or the green cells, they're called secretory cells. They produce mucus. And the ciliated cells, these cells have little hair, cilia, and these hair, they can push out the mucus out of your lung. So, and then you have some basal cells, that sit on the bottom and hold the tissue together, so to say. Now, if you injure the lungs, for example, if you have the flu, maybe also if you have COVID-19 or so, then you lose a lot of these cells here in this basal layer. And what happens is that these other cells, the blue and the green cells, they can rapidly reprogram and turn into new basal cells. So how can these cells at the same time be very robustly specialized over the whole lifetime almost to do one specific job, but at the same time, if you give them the right signal, rapidly program and do something entirely different? So this is the question I would like to address in the first part of my talk. And we asked this question in what biologists call a model system. Yeah, this model system here is called Polistes canadensis. It's a so-called paper wasp. And this paper wasp lives in Panama. And what we understand then from this model system, this biological model system, we can then use to generate hypotheses, and these hypotheses we can then test in other systems like in cells. Yeah, so what are these paper wasps? Yeah, so these paper wasps live in little societies. Yeah, they have a stable social order. One of these wasps is a queen, and the other wasps are workers. The queen is the only reproductive individual, and the workers go out for foraging and so on. Yeah. Now, all of these individuals have the same genetic information, but nevertheless, they show very different behavior. So one of them is the queen and the others is the workers, but they all have the same genes. So how is this social structure established? To understand that, it is always good, if you want to understand something, to perturb a system. Yeah, if you want to understand how a bicycle works, it's good just if you turn the pedals and see what happens. And that's what we also do in these insects here. Yeah, what we do is we remove the queen in an experiment such that only the workers remain. Yeah, and then after we've removed the queen, the remaining workers can reprogram and generate a new queen. And this is what we call plasticity. Yeah, it's similar as in the cells. So here, I hope you can see the video. Here you see our collaborator, Solange Patalano. She's in Panama, and she's approaching one of these nests that you see on the top left-hand side. Yeah, and here you can see how she removes the queen from such a nest. And now only workers are left. And now we see what happens. So after we remove the queen, we can do something very unique and very cool. We follow the relaxation process, yeah, the process back into the steady state of the stable social order, simultaneously on different scales of organization. Yeah, so we measure this on the macroscopic level. Yeah, so that we take videos of these nests, and with these videos we can track insects. We can measure the movement of these insects. We can see how they interact. We can see which insect is in the nest and which one is not in the nest. And then we can go all the way, then we can go to the individual level. Yeah, we can measure of individual insects, the age, the size of their ovaries. We can characterize their behavior. And then we go all the way down to the molecular level. And we take these brains and we do these experiments that I mentioned before with this car. Yeah? We have a detailed molecular profile of these brains in these insects while they generate a new queen. Yeah, and the important thing here is that we can correlate all of these measurements to each other. So we can, for the wasp that we see in the video interacting with another wasp, we can at the same time take a look at the brain and see what are the molecular profiles of their brains. Yeah, and this is not something you can do in physics. Yeah, that's something that an example where biology gives us something conceptual new. Now we can follow this system over time at the same time on the microscopic and on the macroscopic level. Now the way this is done, just briefly, is we take the queen out of the nest, and then we follow different nests over different periods of time on video. Yeah, and at the end of each video, we take all insects from a nest, and we dissect them, and we sequence these brains to get the molecules that are in these brains. Yeah. So now to understand how these insects reprogram and how they regulate their society, we started looking at the molecular level. And on the molecular level, in insects or in general biological systems like cells, if you ask a biologist what is the most important thing, they will say it's the genes and gene expression. Yeah, and that's what you see here on the left-hand side. Yeah, you have a gene on the DNA, and you have thousands of different genes, and it's just as these insects, yeah, roughly like 15, 30,000 genes. Yeah, and these genes here is when it is activated, produces so-called RNA molecules, and these RNA molecules are then translated into proteins, and these proteins then do a certain job in the cell. Yeah, and using technology called RNA sequencing, we can measure the abundance of these RNA molecules in every insect in the nest, yeah, and for every gene. Yeah, this is a lot of information, and the first thing we can do is to ask which genes are different between queens and workers. Yeah, we just take a normal nest and we ask which queens are different. Yeah, here you see on the y-axis, yeah, so every dot is a gene. You know, we measure many genes. Now that's this car experiment, very detailed molecular measurement. For every gene, we can measure its expression in a worker and in a queen, and the y-axis is the ratio of both. The green dots here, these are genes that are activated in a queen, but not in workers. And we call them the queen genes. And now we can ask what happens to these queen genes in the workers that are still in the nest. So in this plot on the right-hand side, on the x-axis, you have the expression, so every dot is a gene again. On the x-axis, we have the expression of every gene, yeah, so this number of RNA molecules in every gene in a normal nest with an intact society. And then on the y-axis, we have the expression of the same gene in workers after the queen is removed. And what you see here is now that all these green dots, these queen genes, they all move to the left. And that means that these workers, when you take away the queen, they all activate the genes that make them a queen. Yeah? And this is quite surprising because, or it can't be everything, because we know that in the end we get a single queen, but all of these workers at the same time, they all on the molecular level look like a queen. Yeah, because they express the genes that make them a queen. Yeah, and that means there needs to be something else on the level of the colony or of the population that breaks the symmetry between these workers and that in the end selects a single queen. So, and what this process might be, you can see in these videos here, in this video here. You see a queen and a worker, and you can see the queen, how this queen attacks the worker. Yeah, and these interactions, these fighting interactions are quite frequent between individuals, and we can quantify them. Yeah, we record these nets on video for long periods of time, and at the end of these videos, we measure the expression of these queen genes in the brains of these insects. And that's how we can correlate the expression of these genes with the behavior of insects in these interactions. And just in a nutshell, to give you an idea, what we found is that the more highly an insect expresses these queen genes, the more often it interacts. Now that's the top plot here. And in the bottom plot, what you can see is that in each interaction, like in this video here, there's a dominant part, now the attacker, and a passive part. And whether you are the dominant part or the passive part in such an interaction, also depends on how highly you express these queen genes. Yeah, the more highly you express the queen genes, yeah, the more aggressive you are in these interactions. And now we can put this all together, yeah, and come up with a simple model and try to understand how the society and the plasticity of the society is regulated. Yeah, so we start on the molecular level. Yeah, on the molecular level, we have these queen genes. And if nothing happens, remember like 2 slides ago, if nothing happens, these individuals just express their queen genes. Yeah, they all want to become a queen. But in the context of a nest, yeah, on the left-hand side, in the context of the nest, these insects interact with each other. And when they interact with each other, they exchange signaling molecules, yeah, probably pheromones, but can also be a different kind of molecule. They exchange the signaling molecules, and these signaling molecules then repress the expression of the very same genes. So the collective behavior on the level of the nest, of the population, influences the behavior on the molecular level, influences gene expression. And as I showed you on the previous slide, gene expression also changes how these insects behave on the population level, how they behave in these interactions. So you see how the system consists of different scales of organization, from molecules to the nest, to the population, and how they transmit information between these different scales. And we can formalize this mathematically. In the end, we're doing a theory. And mathematically, most of you won't have heard of these kind of equations. Yeah? So this is a master equation, so-called master equation. It describes the probability P that that individual I has a gene expression level Ri and a concentration of the signaling molecules Qi. Yeah, and the time evolution of this probability here, it's very high dimensional, is given by this master equation here. It's not so important to look into this, yeah, but we can solve these master equations, first of all, numerically, using stochastic simulations. Yeah, and this is what I'll show you now here. On the left-hand side, if you look here, this is the result of such a master equation, of such a simulation. On the y-axis, you have a histogram, so that the probability that you have a certain gene expression level, R, and from top to bottom, you have the time. Yeah. So if you look at the top row, the initial state of the simulation, you have workers, yeah, that have very low queen gene expression. Yeah, so these are workers that don't express these genes. And then you have a queen here that has very high levels of queen gene expression. And now we take away the queen from this nest, and then we let the dynamics evolve over time. And what you see that these workers, they all upregulate these queen genes in these simulations here. You see that this peak here travels to the right. And at the very end of this process, if you wait long enough, this wave splits up and turns, and you get 2 distinct peaks. Yeah, and that is this specialization to queen and workers that I mentioned before. Yeah, so the simulation is doing what we expected to see. It recapitulates the dynamics in this reprogramming process, and we can then ask, so what is the range of possible behaviors of such a model? Yeah, and this is depicted in the so-called phase diagram. That's what you see on the right-hand side. Now, so here you have 2 axes. On the x-axis is the interaction rate, so how often do individuals interact with each other? And on the y-axis is a measure of how sensitively insects can measure the state of other insects, so the asymmetry in these interactions. Yeah, and what you can see here is that this model shows different kind of behavior. Actually, you have phase transitions in this model. Yeah, if the interaction rate is very low, then all individuals will turn into a queen. Yeah, so that they will all be egg layers, or they will all be reproductive. Yeah, and this is the evolutionarily primitive state. That's how these insects were a very, very long time ago. Now, if you increase this interaction rate, then you get a social phase where you both get queens and workers. And if you just interact often enough, that's this shaded region here, you're always sure to get a single queen. Yeah? You don't need to turn parameters to get a single queen. You're always sure to get just one queen and not two or three. Yeah? And it happens that the parameters that we measure from our experiments, that they just happen to be in this phase where you expect to see a single queen. And indeed, they seem to be achieving this specialization to get a single queen with a minimal effort that is necessary, with a minimal degree of asymmetry and with the minimal necessary interaction rate. So this was all very descriptive. Yeah? I just showed you that this model does what we expect to see in terms of biology. Yeah? So that means that the model is probably right. Yeah, and we of course have undergone all kinds of other tests to show that this is actually correct. But we still haven't understood how these insects are regulating, are being plastic and robustly specialized at the same time. Yeah, to understand that, it is instructive to approximate the equation that I showed you before and go to a certain so-called mean field limit. Yeah, and this mean field limit, you neglect basically stochastic fluctuations, stochastic noise. Yeah, and then the equation becomes sufficiently simple that you can actually understand it. What you see here is the probability f, yeah, that a certain, or the number of wasps f, that is certain that the number of individuals that have gene expression r at a time t. Yeah, that's the black term here. And this number of individuals with a certain gene expression level is determined by two factors. On the left-hand side, you don't need to understand this equation. On the left-hand side, you have a component that describes the molecular dynamics. And on the right-hand side, you have a component that describes the feedback of the population on the molecules, yeah, the population dynamics. Yeah, and if you look at these kind of equations, if you're a little bit advanced in your studies already, yeah, then you might recognize that this is an equation that typically describes as similar than to what describes colloids or something like the Boltzmann equation. Yeah, and you will see that this equation reaches a steady state. Yeah, so the time derivative becomes zero if the molecular dynamics in blue and the population dynamics in green balance. Yeah, and this is the steady state when these two factors balanced. And now it's instructive to look at the co-evolution of both dynamics. And now we get a little bit more abstract. Yeah, now it gets theoretical. So what I'm plotting now is is a so-called phase portrait. Yeah, in this phase portrait on the x-axis is the gene expression state, the molecular state of a single individual. And on the y-axis is the population state, the composition of the population. Yeah, would be this f here. And because we cannot plot a function on one axis, yeah, we'll plot that's just the average gene expression in the society. Now, suppose you put a virtual wasp, into a nest that has a hypothetical state that corresponds to this dashed line. Yeah, then this virtual wasp, this tracer wasp, as we call it, will follow these black lines and turn into one of these stable states. Yeah, so that the nest will evolve to this solid green line, and this virtual individual will follow the black line and take one state on this green line here. Now, in this green line, the steady state of the population, we have, again, these dots here. Yeah, these are stable states. One of them is the queen, and another one is the worker state. Yeah, and now we can ask what happens to the system if we perturb it. Yeah, and there are different kinds of perturbation. One kind of perturbation are intrinsic perturbations. Yeah, so these are perturbations that affect insects individually. For example, gene expression noise, thermal fluctuations, yeah, or the decay of molecules or things like that. And these things are not correlated between individuals. Yeah, they average out on the level of the society. And that means that for these perturbations, you always stay on the green line. Yeah, you only move on the x-axis, but not on the y-axis. Yeah, and if you move on this x-axis and you get a perturbation, yeah, you go to the left or to the right, then these arrows, these black arrows, point into the opposite direction, and you will always get black back into your stable state, that is the queen or worker. So these systems are stable, these insect societies are stable against intrinsic perturbations. And now we can look at another kind of perturbation, namely extrinsic perturbations. So extrinsic perturbations are perturbations that affect the society as a whole. Think about like a signal that they all get at the same time, like a varying temperature, but also perturbations that change the composition of the society. For example, when you remove the queen. And what happens then is that you go diagonally in this diagram, you change the molecular level and the population level at the same time. So both scales of organization are changed at the same time. And what then happens is that you end up on this blue dashed line. Now, this is mathematically called a separatrix. And what that means is that when you are on this blue dashed line, you have arrows going to the left and arrows going to the right. So when you are on this blue line, you have the same probability of becoming a queen and a worker. You can do both. And this is the definition of plasticity. So what this means is that these insects use antagonistic dynamics on different levels of organization, on the macroscopic level and the microscopic level, yeah, antagonistic dynamics. And with this, they're able to distinguish between different kinds of perturbations. They can be stable against intrinsic perturbations, yeah, like intrinsic noise, but at the same time, they can be plastic or flexible with respect to extrinsic perturbations, such as when you remove the queen. And this is how these insects at the same time achieve plasticity and robust specialization. And this is only possible by this very peculiar nature of biological systems, that you have different dynamics on different spatial scales happening. Yeah. So with this, I would like to conclude. There's much more to say about these insects. They're actually pretty cool. So with this, I would like to conclude the first part. You can see how these, I showed you how these insects have the system of sending around information on different scales of organization, on different levels of information, dynamical processes happening and being coupled on these different levels of organization that allows them to achieve plasticity and long-term specialization at the same time, something that is actually on first sight very paradoxical. Yeah, in terms of physics, this shows how, just generally speaking, just to bring a little bit of physics jargon, this shows how macroscopic states can organize microscopic order in general systems. Yeah, and the reason why this work is important, that actually these kind of experiments, they anticipate developments that will come up in stem cell biology, yeah, where also the plasticity of these cells is one of actually the lung examples I gave you, is one of the central questions in stem cell biology. And we're now at the verge of being able to do similar kinds of experiments in these cell systems as well. And then it becomes very important for things like injuries and cancer and so on. Yeah. So with this, I would like to conclude the first part of my talk. Yeah. And I would, so if there is already any kind of question, so just let me know right now. Otherwise, we can answer them all together at the end of this talk. Yeah. So if there are no questions, then let me go to the second part of my talk. Yeah, and now it gets a little bit more biological, because now we're going into the messy stuff that's called cells and tissues. Yeah, and this is, of course, what most biologists are interested in. How do our organs form? How do cells make complicated decisions in development, in aging, in the case of a disease? Yeah, how are these processes controlled? Yeah, and then experimental technologies are much more limited. Yeah, it's not like in the social insects that you can just video record cells in an tissues. You can't put a camera into an embryo and expect that this embryo just continues development happily. Yeah, that doesn't work. So when we want to learn about cells, taking videos, cells in, for example, an embryo or in a living system, taking videos doesn't work because these videos would kill the animal. What we do instead in biology is that we use sophisticated genetic methods to learn about cells. Yeah, and one of these methods is called lineage tracing. So in lineage tracing, genes we can genetically label a certain subset of cells that interests us in a tissue. Yeah, and then these cells express a reporter protein, a fluorescent protein that we can see under the microscope. Now, as cells divide, this marker, this label, is passed on to all progeny of a marked cell. And this progeny is then called a clone. Now, on the left-hand side, you can see such a clone in epidermis, that means in skin. Yeah, and so, for example, here, this clone here would have 4 cells at 3 weeks. What is important to understand that also here, once you want to look at these clones under the microscope, you still have to dissect the animal. So the animal still dies. Yeah, it's a little bit like quantum mechanics. You make a measurement and the wave function collapses. Yeah, so same here. Here in biology, once you make a measurement, yeah, you have to kill the animal, and you can't do the same measurement again in the same animal. But what you have here is that you initially label one cell. Now you label one cell, and then you look at a later point or a subset of cells. You label them randomly in the tissue. And then a later point, when you analyze, when you dissect the animal, you see a collection of cells, of these clones here. So this is a dynamic measurement. It's a poor man's version of a clone. You have 2 time points, one cell at the beginning and X cells at a later time point. It's dynamic information. It's 2 frames of a video at least. But 2 frames is already enough, because the sizes of these clones here, they carry information about what cells have been doing between labeling and analysis. And if you think, people think that if you repeat this experiment here often enough, and you get large statistical ensembles of these clones, then you can use methods from statistical physics and statistics to reconstruct what happens between labeling and analysis. That was what people are doing in the field. It's the working horse, so to say, of stem cell biology. But there is a problem with this method. Namely, that when you, especially for example, that what you have in dynamic tissues, like in a tumor or in a developing embryo. So these are very dynamic tissues, and you have random forces all the time in these tissues when cells push against each other. What happens then in these tissues is that these clones don't stay cohesive all the time. There are these blobs that you saw on the last picture. But these clones, because of all these forces, fragment into disconnected clusters. And for the very same reason, initially separated clones can merge and form larger compounds of labeled cells. Yeah, in the end, it's pretty messy. Yeah? It's a soup of cells of these clusters, and here you can see fluorescent images on the right-hand side from the heart tissue, from a heart experiment, and on the left-hand side from a pancreas experiment. And you would have a hard time understanding what is a clone and what is fragments from the same clone here. Yeah. So what can we actually learn from these kind of experiments about what the cells are doing during development? This is the question I'd like to answer in the second part of my talk. And we started thinking about this question in the context of mouse heart development. Yeah. And in this experiment, together with a group in Brussels in Belgium, this group labeled the earliest founder cells of the heart in a mouse embryo. Yeah, roughly 6 days after fertilization, you can identify the first cells of an embryo. Yeah, and then you label these cells with these lineage tracing experiments. And at later stages during development, you look at these, you take the hearts out of these animals, you put them under the microscope, yeah, and you can see on the right-hand side these clusters here on the surface of the heart. Here are 2 time points. The upper time point, E12.5, is early development, yeah, embryonic day 12, and the lower picture was taken at the date of birth of the mouse. Yeah, you see these patches on the surface of the heart, and now you can go on and measure the sizes of these patches. Now you can do that automatically with certain software that you can outline the boundaries here of these clusters, and then you can measure their sizes. Now, if you go on, yeah, so you measure these sizes, and what you then get is a distribution. Yeah, you get probability distributions of these sizes. Yeah, these plots are just the way that biologists plot probability distributions. Yeah, every dot here is one of these clusters. And we did these experiments not only for heart, but also for other tissues, for example, liver development with the lab of Mary Hook, and pancreas development with the lab of Anna Philpott in Cambridge. Yeah, so it's a very similar experiment that people use in a very different context. Now, when we looked at this data for mouse heart development, we were surprised that it had a very simple mathematical structure. On the top left, you can see that the average size of these clusters is very different between the different regions of the heart, the different bars here, and different time points in development, the different colors. And this is, of course, that the average is different is very expected, because we know that every individual cell follows a very complicated developmental program. Yeah, this is completely expected that what we get is also complicated and reflects this complexity of embryonic development. But what we found is that once you divide the size of every cluster by its average, then these probability distributions perfectly collapse, they perfectly overlap. So here in this plot, you have a cumulative probability. That means, for example, here that roughly, what is it, maybe 10, 15% or 10% of these clusters have a size larger than 2 times the average. That's how to read these plots. And these distributions, once we take away the average, they perfectly overlap. So that means once we know the average of these measurements, we know already everything. There's not more information. And this is what we call in physics scaling behavior. Yeah, so once you know the average, you already know everything. Yeah, and this observation of scaling behavior suggests that there's a simple mechanism that creates these simple distributions. and this mechanism could also be more general than just for mouse heart development. So what could determine this simplicity in the outcome of this experiment? Yeah, it is instructive to think what are actually the processes that determine the sizes of these patches on the surface of the heart. Yeah, and then you will see that there's a very close analogy to what droplets do in aerosols. Yeah, so in aerosols, these little droplets, they can coalesce, they collide, and they can form larger droplets. Yeah, and droplets can also take up water molecules from the environment, from the vacuum, and grow by taking up these molecules. These droplets can shrink by evaporation. You can have nucleation of new droplets from single water molecules. So water molecules can start growing new droplets. And these droplets can become unstable and fragment into smaller droplets. Yeah. So, and there's a one-to-one analogy between what determines the dynamics in these aerosols and what determines the clusters in lineage tracing experiments. So growth by condensation of water molecules would correspond to the growth of these clusters by cell division events. In both cases, you have merging and fragmentation of these entities. Nucleation of new droplets is something that you get when cells migrate into the field of view of the microscope, and they only then become visible. And evaporation, so that water molecules leave these droplets into the vacuum, corresponds to cell death. So there's this one-to-one analogy, and I would show you that if it wasn't mathematically actually correct, this analogy. Yeah. So and because of this analogy, we can make use of roughly 100 years of theoretical research on these systems. And the first person who studied such dynamics, like in aerosols, was Mariam Smoluchowski. He was the founder of this theory, and he wrote down equations of the form that you see here. Yeah, this is an equation. It's not so important what the terms in detail mean. It's just the time evolution of the number of droplets that have a certain number of clusters that have a certain size x at a time t, and then this number of droplets are this time evolution of this quantity is determined by many different processes. For example, growth by cell division, fragmentation, merging, and many other biological processes. Now, the kind of equations that you see here is almost impossible to solve. That is too complicated. Only for a very few special cases you can solve these equations. But what we're interested in here in this context is not necessarily the precise solution of these equations, because we can't measure everything in these experiments. Yeah, we have a, so to say, a fuzzy view of these clusters of the cell. We have to zoom out of these clusters in the heart. We have to zoom out to see them. And actually, what is not so important is what single cells do. We don't see that in the experiments. So we ask here, what is actually determining the large scale variability of these clusters? Yeah, what is important on the macroscopic level of the organ? Yeah, and in statistical physics, we have developed tools to just answer this question, and this tool is called renormalization group theory. Yeah, I don't show you the equation, I just show you how this works in principle. So you start with any complicated biological model that has all of these different processes as different terms in them. Yeah, and what you then do is you coarse grain step by step. Yeah, you go to larger and larger scales. You zoom out more and more until you've reached the entire size of the organ. And what you do that on different zoom levels, different processes will describe the behavior to different degree. Different models will describe the behavior of these clusters to different degree. And what we found is that if you do these processes, what survives on the scale of the organ is only merging and fragmentation, and all other processes become involved, become irrelevant. Now, that means whatever you start with, any complex biological model, all of this information will be erased, and only the physics of merging and fragmentation will matter. Yeah, and this is actually an example of a critical phenomenon. If you look at it in detail, and maybe some of you already have heard of critical phenomena, and this criticality, this critical process leads to the erasure of biological information. This biological information statistically does not matter anymore once we zoom out and we look at the whole picture on the size of an organ. Yeah, and this erasure of information, this is actually what tells us why these distributions that I showed you previously, why they take such a simple form. It's just that this complexity that is governing on the small microscopic scales does not matter on the large macroscopic scales. And we can then also make another prediction, namely that actually the distribution that we get, if only the physics matters and not the biology, then this shape is independent of the specific biological context, and we should get the same behavior in other tissues as well. And finally, we can then do more calculations and make a prediction about what this shape of these distributions of these cluster sizes should be. And they take something that is a log normal form, that is like a normal distribution of the logarithms of these sizes of these clusters. Now let's go back to the experiment and see if we are actually right. Yeah, so here you can see the data from mouse heart development. On the left-hand side, you can see again cumulative probabilities. Yeah, the gray line is theory, and the red or blue lines are the experimental predictions. And what you can see is that we predict that with this simple theory, the experimental outcome of the sizes of these clusters extremely well over several orders of magnitude. And we can do that for both time points in development. And if you're really skeptical, then you can also look on the right-hand side on so-called quantile plots. And just in a nutshell, the fact that these are on a straight line show you that this is a very sensitive test, that you predict something very nicely. And the fact that these are straight lines here shows you that we exactly have the right prediction. And that's actually something surprising, because in biology is always complicated and messy. As a physicist, you always have to make approximations. Yeah, you never get perfect fits. But this looks just too perfect for a biological system. And the reason for this is this powerful concept of universality that you will have also in your studies at some point. Yeah, that different a large class of different microscopic rules or behaviors can lead to the same outcome on the macroscopic level, in this case, on the level of the tissue. So now we can go on. Another prediction was, if this is correct, that we should expect to see exactly the same behavior for different tissues as well. So we did all kinds of experiments and collected data from mouse heart, from the liver, from the pancreas, even from other species like zebrafish, and measured the sizes of these clusters that you get in these experiments. And what we see is that they all, once you rescale, once you divide by the average cluster size, they all show the very same behavior that is predicted by the theoretical curve, the dashed line here. So actually, in these experiments, there's no biological information. Yes? The liver looks just like the pancreas or the heart, yeah, and even the zebrafish heart is exactly the same. Yeah. So this is just a reflection of the fact that this universality erases biological information. Now, this is not something that biologists like to hear, yeah, because they're doing these experiments all the time. But on the positive side, understanding what is actually the expected outcome in terms of physics of these experiments, what would you expect as a baseline? Yeah, this is necessary to understand what you want to identify as an experimentalist if something interesting happens in your experiments. Yeah, and to show you how this can work, yeah, we actually have one example here in this inlay of a tissue that is not following the behavior that we predicted, that is not universal. This is a certain class of cells in the pancreas. On the right-hand side in yellow, you can see an example of these cells. Yeah? And if you look at these cells under the microscope, then you can see that these cells migrate along a network, and then they form little cauliflowers at the tip of this network. Yeah, and these little cauliflower-like structures, they're very strongly adhesive. So these cells stick together, they're like glued together. And because they're glued together, there's no fragmentation going on. Yeah, they just stick together and form large balls. And that's why they break this erasure of biological information. Yeah, they don't show the same behavior as the other universal experiments here. And we used this behavior that we would expect based on the physics of the statistical physics of this system to identify the behavior of this cell subpopulation here, which is in the end a very important process in embryonic development. So just to conclude the second part and last part of my talk, so I showed you that these clones, these clusters on the surface of these developing organs shows this simple behavior that is characterized by these scaling distributions that are almost void of information and that are also universal. That means that there is no difference between different kinds of biological tissues. And this is very surprising. Yeah? So we know that these cells are doing complicated things. We know that already. So every cell is doing a different complicated program. Yeah, that's almost deterministic. We know that. But once you put these cells together in these clusters, you get something that is almost void of information. And that's a hallmark of critical processes. Yeah, and in this part here, in this kind of experiment, it's very important to understand these processes because otherwise you cannot interpret these experiments. And what I think is also cool is that we do physics, yeah, and at your later stages of your study, you will learn about criticality and universality, all these very central concepts in theoretical physics. And then you look at an embryo and you find the same concepts popping up. Yeah, and this is just for me an example how powerful some of these concepts are that we developed in statistical physics or in theoretical physics in the last 50 years, that you again and again in completely unrelated examples, they come popping up and they're proved to be essential just to understand these biological systems. So with this, I want to conclude. And before I conclude, I wanted to show you, if time permits, I wanted to show you just very briefly why it's an exciting time to do statistical physics or theoretical physics in a time where biology is changing rapidly. Yeah, and what I want to say is that there are very striking differences, yeah, in condensed physical matter and living matter. And these differences are in such a way that we need actually new theory to describe the biological systems efficiently. Yeah, and one of the striking differences is that physical systems are defined by symmetries, while biological systems on the microscopic symmetries and living systems are microscopically very, very messy. Yeah, this is already where it starts, where you have problems just describing these kind of phenomena. And this poses very challenging questions for non-equilibrium physics, for theoretical physics in general. And it's, I think, a great time to be going into theoretical physics and to try to address these very fundamental differences between physical and living matter. Yeah, just let me thank a bunch of experimentalists, Wolf Reich in Cambridge, Solen Patalano, who did these experiments on the wasps, and a lot of other people who contributed to lineage tracing, and Adolfo Alsina, and Misha, who did the theory for the wasp projects. Yeah, so now I'm a few minutes over time, I think, and rapidly wrap up. Thanks for your patience, and happy to take questions. Thanks a lot.
Thank you so much, sir. That was a very enlightening talk. Now we are ready to take questions from our audience. Okay. So we have a question from Bhavneet. He asks, what happens when we add a queen to a system with two distinct peaks at gene expression level, assuming both the queens are equally strong? And can this be simulated as an extrinsic perturbation?
Yes, that's a very interesting question, and we can simulate this. And this system allows only, so from the theoretical part, this system only allows for a single queen. So one of these queens will become a worker. But if you do the same thing in experiments, then it's more complicated. Yeah, so then these individuals, these insects are more than the particles that we physicists treat them as. And what happens in the real biological systems, you get these two queens, they will fight all the time, it will be very aggressive. And at some point, one of the queens leaves the nest and founds a new nest. The end result is that you, in the end, only have a single queen all the time.
Another question from Kunal. Is there an upper limit on the society size that a queen controls? 5 wasps or 10 wasps? And how can we model this?
Yes, we actually modeled this, so I didn't show you that. The larger the size of the society becomes, the more busy the queen will be controlling the workers. Yeah, so it's very, in principle, it's quite simple. Yeah, so a queen cannot interact with 100 workers all the time. Yeah, and what you will need is that there are additional processes happening. Yeah, and that's actually some work we did. There are so-called epigenetic processes that help the queen controlling the society and controlling the noise in the society. Yeah, but interestingly, in nature, if you observe a nest for a long time, it will grow and grow and grow. But at some point in time, the queen will get overwhelmed. Yeah, and what then happens is that the nest becomes unstable and falls apart, and these insects, they will find new nests somewhere else. Yeah, so this nest becomes unstable if the queen cannot control it anymore. A very nice question.
Another question from Abhas. Is universality a test for designing models, or do we test for universality in our models? Like, confusing. Abbas,
yes, I think it's the latter one. Yeah, so very few models give universality. There's no reason why universality should pop up in biological systems, although we see it many times. Universality is something that you see a lot, like in phase transitions in magnets and so on. If you do statistical physics, you have your first statistical physics lectures, you will see that universality goes along with phase transitions in these systems. We don't have a general theory for universality in systems that are out of thermal equilibrium. Yeah, so but we see that all the time. There are some theories about where it comes from. Yeah, but it's in the end, it's something that pops up surprisingly for us, like in the system that I just showed you. But we wouldn't design a model from the start to get universality. Yeah, so here it was a prediction, that we then tested in experiments, in other experiments.
Sir, I have a question. So can we compare biological stochasticity with the plasticity, like how it affects it?
Yes, actually, that's also something we did. And what we found is actually, so first of all, you would think that if this society, these systems are always noisy. Yeah, if you only have 10 insects or so in such a small nest, then it matters what a single insect does. Yeah, so that's like you take one insect out and it's already a 10% difference. It's not like in a gas or in a physical system where you take one molecule out, and it doesn't really matter because you have 10 to the power of 26 other molecules. Yeah, and these kind of small biological systems, this noise, this stochasticity matters a lot, and these insects actually have a problem controlling it. Yeah, that's one way, but they managed to control it using additional molecular processes that I didn't show you. Yeah, they managed to control it. But what you would think is that now you've got your stochasticity under control. Now you're super stable. But what you then typically have is that if you suppress noise, if you lock your system in, then you also have a very difficult time in changing your behavior, your plasticity. Now, this is exactly this paradox that is out there in the field, that how can you be at the same time stable against this noise, not have much noise, but at the same time also then be very flexible to very specific signals. Now, that's why you have to distinguish, is it something, what I'm seeing here, is it something that is noise, or is it something that is a real important signal? That's the key challenge for many biological systems, also cells. And it's always a puzzle of how they get along, although in biological systems, a lot of time, noise is even stronger than the actual deterministic effects, than the actual average that you want to achieve. Yeah, so they always very often dominated even by noise, and they develop strategies to make decisions despite of strong noise. Yeah, I hope this answered the question. I'm not sure I completely understood it.
Yes, clear. So we have one more question in the comments. If you allow us, if time permits, we can have that.
Sure.
Yeah. So another question from Anurag. He asks, can you comment on the erasure of biological information in the context of benign tumor, where fragmentation isn't seen? Does it then possibly break the law of universality, similar to the ACNR cells?
Yes. So if you don't have fragmentation, then you don't have universality. Yeah, if you have fragmentation, a lot depends on the tissue. And it's very hard to show that you don't have fragmentation. Yeah? Even in very steady tissues, like the brain, or so that grow very slowly, even there you have fragmentation, although you don't see it at the very first time. It's not as extreme as in the heart. Yeah, for tumors, I don't know. We didn't have the data on this time. But I would expect that if you have fragmentation, then you have universality. If you don't have fragmentation, then you could still have universality, but a different kind of universality. Yeah, it could still be universal, but you would have to check then in this specific case.
So we have no more comments now. So I would like to wrap up this webinar. So thank you once again, Dr. Rulands, to be here.
Yeah, thank you. Thanks for the invitation, and wish you all a lot of fun in this Schrodinger week.
Thank you so much, sir. And thank you to all our audience who attended this webinar, and stay connected with us on our website and for the coming events. So stay home and stay safe. Thank you so much.